that the time is ripe. We are at a point when people are more willing to make this bridge between a memory of a human, a memory of an animal, and a memory of a cell. The fact that patterns matter on a finer time scale that we previously believed makes it possible that in the future we will be able to construct our experience based on what we know about how those patterns converted to what outcomes. I know it's very general at the moment. I don't have a specific technique to save yourself from Alzheimer's, but I think those techniques might exist, and it's up to us to find them out. And that's a big part of our message to the world, that when we're saying cellular memory or cellular cognition, we're not using these words in quotation marks. We mean them literally. We mean that the more advanced forms of cognition and memory are built upon these molecular tools that have been around since before there were brains and animals and humans and anything like that. So all of these processes have deeper evolutionary roots than we like to believe.
Welcome to the FitMind podcast, where we explore the frontiers of human potential, meditation, and neuroscience. What if memory isn't just in your brain, but in every cell of your body? Today's guest, Dr. Nikolay Kukushkin, is a neuroscientist whose groundbreaking research reveals that even individual cells, like those in your kidney or skin, can learn from patterns and store memory. We explore what this means for trauma, behavior, consciousness, and even the future of AI. We also touch on how this cellular intelligence mirrors ancient contemplative insights, where the body plays a central role in emotional healing and awareness. This conversation is a deep dive into the biology of experience, how life remembers, and it might just shift how you think about who or what you are. I'm your host, Josh, and I hope you enjoy this episode. Nico, welcome to the podcast.
Thanks. Nice to be here, Josh.
Great to see you, and I'm really excited to chat. So you've had an extremely colourful background, I would say. I'd love it if you could just share a little bit about your background and how it was that you got into neuroscience, in particular the study of memory and the evolution of the mind.
Yeah, sure. So my background is in biology. I'm originally a biologist. Anything that I think about in the brain, the first thing I think about is evolution. I don't think about what happened to me yesterday. I think about what happened to my species a billion years ago. That's my frame of mind. But when I first started in biology in college, I really didn't have any idea of what I was going to do, that I was going to be interested in neuroscience. None of that was clear to me. The way I thought about this originally is I wanted to do the big medical problems, solve global diseases, cancer, HIV. I think that's what most people start with. And so I'm from Russia. I like to say that I'm from both a city and a country that no longer exists, actually. I'm from Leningrad, USSR, which is an interesting fun fact. But I went to school in there, to undergrad, and after that, PhD in the UK, in Oxford. And that's when I started thinking more about biochemistry, smaller things, cell biology. And my PhD project was about how molecules in the cell talk to each other. So it wasn't about the minds yet, and nothing to do with memory, but it was about information processing. It was about how molecules inside the cell coordinate their own activities to produce proteins for export, for example. And there was some clever information processing going on between them. Then I did my first postdoc, which was still in that line of work, still about cancer, but also a little bit about dementia. Since we were working in the way that proteins are being produced, it goes wrong in cancer. It also goes wrong in some dementias like Alzheimer's, Parkinson's, frontotemporal dementia. There's some common threads between those different diseases. And so that was the first time I touched anything to do with memory. It wasn't yet about how memories are formed, what kind of information processing goes on in the brain. It was more about what breaks down in cells, in neurons during dementias and these diseases of the brain. And from there on, I became more and more interested in the question, okay, what happens in health, in normal brains when we form memories? What happens to those cells and molecules when we learn, when we try to memorize something? It's this connection between the molecular and the profound, the philosophical, the mental, that's most interesting to me. That's what I'm always looking for. I'm curious about how we get from chemistry, from the smallest components of a living organism, to our own minds, to our own internal perspective. So from there on, I went to the lab where I currently work. It's Tom Caru's lab. Tom Carew is my mentor, and my everything I know about neuroscience I know from Tom. And Tom's lab has been studying Seaslugs for decades. That's what it's famous for. So I joined Tom in studying the Seaslugs. We studied memory in Seaslugs for several years. It's not obvious to outsiders why you would go to Seaslugs if you want to study memory, but actually, it's a very common research model for memory studies because Seaslugs are such simple animals. And not just simple, when you say simple, it seems like this, it's something derogatory that we're normal and they are stupid and simple. Actually, they are normal. Their level of simplicity, that's the standard animal organism. That's the normal animal brain. We are weird. We have a lot more going on that's very hard to understand. But sea slugs is your generic animal. And so I was interested in memory as a concept, as in principle. And for that, you don't want all this complexity that comes with a human or even with a mouse. You want it as simple as possible, simplest memories, simplest reflexes. That gets you to what is happening inside cells, inside neurons, when those memories of a sea slug are being formed. So we studied sea slugs, simple properties of memory formation. For example, one of the studies that we did was we found out that neurons, individual neurons, no complicated arrangements of those neurons, nothing complex about the brain itself, but individual cells of the sea slug can distinguish between a series of events that are escalating in intensity compared to that same exact series of events that's waning in intensity. So the only difference is the order of events. The overall intensity is the same, the nature of those events is the same, but a neuron changes itself more strongly if the series is escalating, which makes sense, for whether it's a bad event or a good event, whatever it means for an animal, you want to respond to it more strongly. You want to change yourself, form a stronger memory if it is increasing in intensity. If it's increasing, well, it's going to be more relevant to your life in the future. If it's waning, maybe it's not as important anymore. So it's a very simple form of pattern discrimination that even an individual cell can do. So that's what I was doing for a few years in Tom's lab, and from there on, you can see that it's a natural question to ask. Okay, if a single neuron can tell apart patterns, well, does it even have to be a neuron? Maybe all cells can do that. Maybe even generic non-neuronal cells can tell apart different patterns. Maybe they can tell time. Maybe they can tell repetition versus the same number of events compressed, crammed in one go. And that's exactly what we found when we looked at non-neural cells, and that's our latest interest. That's what we've been studying for the past couple of years. Non-neurons. We use neuroblastoma cells that are kind of undifferentiated neurons. We use kidney cells that are even more removed from the nervous system. It doesn't even matter which cell you use, they all behave the same way. They can tell apart patterns. We administer experiences, we can call them experiences, but what really are chemical pulses administered at different times. We use chemicals to imitate what those cells might be hearing, what kind of signals they might be receiving from other cells. So instead, we deliver those signals artificially, but we can control with what intensity we deliver those signals, for what time, in what order, and by doing that, we can then monitor how the cells respond. So basically what we found is that surprisingly, even very short stimuli, minute scale time patterns, have major effects on these cells. And what's most interesting is that these generic cells, nobody thought of them as being smart. We always thought of neurons as being smart, and generic cells, well, they can't distinguish apart anything that happens on the scale of minutes. But it turns out that they can. It turns out that they know the difference between four chemical pulses separated by 10 minutes. 10 minutes is really nothing for a generic cell of the body, separated by 10 minutes versus those same four pulses crammed into a single 12-minute long pulse. They know the difference. They know that four pulses separated by time is a more salient stimulus, that they need to change themselves more strongly. That's our latest study published in Nature Communications in November last year. So basically the bottom line is all cells can tell apart patterns. Patterns matter, and they matter even on these very fine time scales. That's where we are. That's my life story.
That's incredible. Thank you so much for sharing. Is I mean, that feels like a paradigm shift where, like, where do you even put that in terms of it feels like biology is kind of merging with neuroscience? I can imagine both would be
Yeah,
extremely interested in that sort of
definitely.
finding. How are you finding the community to react to that? How do you measure the intensity, or how do you
Hmm.
increase the intensity? And how can you tell how the cell is reacting to the time
Hmm.
differentiation?
Hmm. Hmm.
Sorry, a few questions
Yeah,
there.
no, a few questions. Let me address the technical questions first, and then we can talk about what the conversation around this is. So, how can we tell? So, first of all, I have to say that these cells that we're studying, um, they are artificial in that it's a reporter system. We've created these cells specifically to report to us if their memories are being formed. We don't yet know what those memories of those cells actually represent in a living organism. Just to give you a sense of what that might be, let me give you some examples that I know could be true from other research from the past. For example, the example that I always use is we have cells in our pancreas that produce insulin. The hormone insulin responds to food. It helps us absorb nutrients when we've eaten a high calorie meal. We release that hormone, the glucose, sugars get absorbed into tissues. That's what it does. So the more you eat, the more insulin you should be releasing. Let's say you've consumed an extremely large piece of food, something very sweet. You've maxed out on like your sugar intake. And so you've loaded up this sugar into your bloodstream and your pancreas has released a burst of insulin. Okay, so that's the response of those cells. They see glucose, they respond with this burst of insulin. Now you wait 30 minutes and you repeat the same thing. You once again load the bloodstream with the same amount of glucose, overloaded. But now those same cells release twice as much insulin. So you've maxed that out the first time, they released this burst. Now you've waited and they've adjusted themselves, and now they release twice as much. It makes sense. If you've maxed out on your sugar absorbing capacity, you would want to ramp it up because well you don't want to you want to take advantage of all the nutrients that you have. If you've maxed out, well, let's increase it so you can absorb all that glucose, but you don't want it to constantly be ramped up because if you're producing too much insulin you'd just be hungry all the time and fatigued. Probably it would also not be productive. So this ability of these pancreatic cells to adjust their insulin responses to glucose, that's memory. That's and it's useful for the same reason that our mental memory is useful for us. It adjusts us to ongoing experience. So something like that would be body memory of cells around our body. It could be similar in kidneys, it could be similar in our gut, in our skin, in our bones. All of those cells have their own experiences. For the pancreatic cells, it's the experience of seeing sugar. For cells in the gut, it might be nutrients passing through the gut. It might be for kidney cells, it might be salts and fluids. It's signals from other cells. All cells are constantly exposed to this pattern of signals that are coming from all directions. And those patterns could change those cells in ways that will later adjust what they do. Precisely what kinds of changes happen in what kinds of organs, we don't yet know. That's a whole new area of investigation that's now open. But what our cells show us is that fine inputs get converted into long-term outputs. So what are the inputs and the outputs? Artificial. The inputs are molecules, chemicals that we artificially put in that we know we picked those chemicals. Why did we pick them? Because we know that they activate in those cells the same molecules that activate in the neurons when we learn things. We already know this. We know enough about memory to know what happens when you're studying for an exam. This and this molecule gets activated in your neurons. We said, okay, what if we just directly activate those same molecules in other cells? If you imitate this learning, but in other cells. So we can do that with artificial molecules. We can basically reach into those cells and turn on those same components of the cells that are turned on in neurons. We don't yet know what turns them on in real life. We know that it's possible, but we don't know what in our bodies would be doing the same thing that we're doing artificially. And then the output of those cells, it's also artificial. Again, we know from neurons, we know that there are some genes that are we call them memory genes as a shorthand, that are activated in neurons when we form a memory. So for a neuron to form a memory, it has to turn on this gene and this gene and this gene. And when they are turned on, they produce proteins. Those proteins travel around the cell, they restructure its synapses, they change the cell, and so the cell gets modified. So you need to turn on those memory genes. And there are some common genes that get turned on anytime a neuron forms a memory. So we also know that. And we can also find those genes in those cells. So what we did was to install this artificial gene that basically has the same on button as those natural memory genes, but instead of actually producing something useful, it's producing this glowing protein that has no purpose in our cells, except it tells us that the cell has turned on its memory gene. So the cell literally starts glowing. This protein is taken from a firefly. It's called luciferase. It's a common thing to use in biology. It's a glowing protein that you can install in your cell. When you activate the gene that you're interested in, the cell produces that protein, and you can detect light coming from those cells. So literally, we have these cells, we're putting in chemicals, and we're getting light back. That's our experiment. And we have these paradigms. We call them paradigms. They're like musical scores, basically.
Wow.
So you see here each of these has 6 lines. That's because our petri dishes have 6 wells. So each of these wells receives a particular pattern of chemicals. So you see 4 pulses separated by 10 minutes. And right now we're testing what happens if you put another chemical an hour before that or an hour after that. Here is another plate that we stimulate in a different time, and so on. So for every experiment, we have a map like this. And then when we do it, we just add these chemicals going from left to right here, in this particular. It's like playing a piece on a musical instrument. I love that there are 6 lines because it's literally like a musical score. I just love that, but it's a
Right.
total coincidence.
That's
Yeah.
amazing.
So your question was, what do we put in? What do we take out? That's what we do. So it's an artificial system that just tells us that these cells can tell apart patterns, and so if they can, cells of the body can as well. Your other question was, what do the biologists and the neuroscientists think about this merger of the two? Honestly, I think that the time is ripe. I think that we are at a point when people are more willing to make this bridge between a memory of a human, a memory of an animal, and a memory of a cell. It's a It seems like this would be a trivial question, but actually we've had a lot of conversations, serious conversations since this paper came out about whether or not the word memory should be used in quotation marks when we refer to memory of cells. Well, and I always fight for using it literally because well, I don't see any reason why this would be called memory, whereas the memory that our neurons form would be called real memory. And there's no sense that I can discern that makes neuronal memory more real than this memory. It's just our subjective bias. We tend to think that the real memory is what happens to me when I think back to my day, when I think what happened yesterday. That's the real memory, and everything else is memory in quotation marks. But I've worked with Seaslugs, and for a Seaslug, well, a Seaslug does not think about what happened yesterday. A Seaslug does not have our awareness. So if you are going to use the word memory in quotation mark for these cells, well, then you also have to use it in quotation marks for Seaslugs. And if you're going to use it in quotation marks for Seaslugs, you probably should also be using it in quotation marks for mice. And if you're using it in quotation marks for mice, then what is really real memory? So anytime you try to find this dividing line when quotation marks stop being quotation marks, you can't find it. So I think it's an important point that yes, we like to believe that there's something more to our memory, that it's somehow more real, that there's something on top of it, something that finally converts all of these cellular molecular processes into our mind. But there isn't. There isn't. It's just a level of complexity. This is a very simple level of complexity, a very low level of complexity, and in humans it's very high. In Seaslugs, it's somewhere in the middle. That's how I think about it.
It's so interesting in that respect. It might also merge with, obviously, there's a lot of discussion about consciousness currently. Like, where does
Mm.
consciousness, is consciousness
So it,
fundamental? Does it come from matter? Like, Anaka Harris has just put out
mhm,
a really good docu-series for anyone that's not checked it out, where
I've
she
heard about
talks.
it. Haven't seen it yet. Yeah,
It's really good. I haven't listened to the full
uh,
thing yet,
yeah.
but yeah, she obviously did Conscious, a brief history of consciousness
Hmm.
a few years ago as well, and seems to have become even more convinced of that sort of hypotheses. And if that
Hmm.
is the case, well, there's obviously discussion of there's some sort of proto consciousness in cells
Hmm.
potentially.
Hmm.
So how do you define memory? Because obviously that would be defined different to the
Hmm.
zeitgeist or how we think about
Hmm.
memory. So how do you define it like within the lab?
So I define memory as so there's a distinction between defining memory in general and defining, say, neural memory. That's a more specific kind of memory. But memory in general, I define as any quick, no, any 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 time that a quick pattern of inputs gets converted into a longer term output. When quick things cause slow things, that's basically what I call memory. That it's not a response that happens while you're doing something that disappears when you stop doing it. It's a response that's slower than what you're doing and that persists after you stop doing it. And that applies to not just biological organisms. It applies to lots of things. It applies to a piece of dough. If you knead a piece of dough, it becomes more elastic, and that's a memory. It's memory of this viscous piece of matter. It doesn't have to be anything complicated for that. And so I think that is true for cells. It's long-term changes in response to short-term patterns of stimulation. It applies to Seaslugs. It's long-term changes in their behavior in response to short-term stimuli. It also applies to how humans learn. It's long-term changes in our mind and our brain in response to short-term training or learning, exposure, experience.
So there's a lot of, and this has come up a lot in meditation circles, that well, there's a lot of emphasis on the body actually, and some of my teachers in the past have actually implied that, that the trauma is stored in the body, and I think even within psychology that's been pretty well known.
Mm-hmm.
Not to jump to knowing that memories might be stored in cells or are stored in cells, but if it isn't exclusive to the brain, what does that mean for how like trauma and stress is stored in the body? Is that something that has been looked at yet, or is that a little
Mm-hmm.
bit further down the line?
It is further down the line as almost anything, since our study is basically just a proof of principle. It is really, square one into trying to understand what cellular memory might mean for everyday life. But it is interesting that you ask this question because it is something that, as we were working on this study, did not even cross our mind, and yet it was the first thing that we've heard when the study was released. It was the number one commentary coming from all directions, and it made me really think about it seriously. When I first heard this idea that that kidney cells have memory, therefore your trauma might be stored in those kidney cells, I thought that's not what we found. That's not what it is about. Kidney cells have kidney memories. It's not that kidney cells store the brain memories, and trauma is still a brain memory. But then, as I kept thinking about this, um, I've changed my stance. I don't think it's as crazy as I initially may have thought. And actually, I think it maybe is quite likely that there is a mutual connection between emotional traumatic experiences and the body. We know that the brain can have an effect on the body. There's lots of chemicals, there's lots of signals traveling from the brain to the body. Cortisol, for example, a stress hormone, typical simple example. It definitely affects things in the brain, it affects our mental processing, it also affects our body. So, it can easily stand as a communicator between what happens in the brain and the body. We also know that the body has the effect on the brain. For example, gut bacteria could have an effect on anxiety. Something that was considered insane 20 years ago, but now it's an established fact. So both directions exist. And since we know that the body can store in its state, in its configuration, patterns of experience, well, it's not impossible that those fine patterns involve, for example, responses to cortisol. Just one example, but there could be thousands of others that the cells would be able to change differently depending on the pattern of exposure to the stress hormone. And maybe these changes would then feed back on the brain and change the emotional state in one direction or another. Now, I don't have any evidence that that is the case, but I don't think that this is unprecedented or unlikely even anymore. I think it is quite likely that the processes that we know about in these cells are involved in some mind to brain communication. And yes, maybe it has to do with the body keeping the score, with trauma having effects beyond the brain and being hard to treat with only the focus on the brain. Maybe the focus on the body in treatment of trauma relates to these cellular memories. I think that's a very possible um, very, very reasonable theory.
Yeah, regardless, subjectively, it feels that way. Like
Mm-hmm,
the
mm-hmm,
the feeling of often the feeling of anxiety will be around the sort of diaphragm, and
mm-hmm,
there's
mm-hmm,
if you do allow that, you back off, allow that to come in, then it does eventually dissipate, and then
mm-hmm,
often might not return, or it could transform into excitement when you don't have this resistance or tension, and then eventually it becomes empty, or
mm-hmm,
it will disappear into no singular object, to
mm-hmm.
use some sort of Buddhist terminology. So
Yeah,
yeah,
it can be hard to tell, we because our perspective, our introspective point of view is embodied, is located at least predominantly in the brain. It can be hard to tell, is this a sensation in the brain or is this a sensation in the body? One example is muscle memory. We call it muscle memory because it feels as if your muscles were memorizing the piece on the piano by themselves. It feels as if your fingers are moving on their own. It feels like you're riding a bike, your muscles are doing it on its own. But it's not your muscles remembering it, it's still your brain. It feels this certain way. So it might be that there are similar interoceptive parts of our brain that give us a sensation that it's this part of the body or that part of the body that is changing or that is memorizing something, while in fact it might be still just a part of the brain. But the other alternative is also possible. It's just hard to parse those things based on your own subjective feeling alone.
definitely, as you say, it seems. I think there might be something there, actually,
Mm-hmm.
from my completely elementary education in biology and neuroscience. But yeah, it's interesting. I often look at the Eastern traditions, and we've proven a lot of what has been talked about or theorized and practiced for thousands of years, potentially up to 10,000, maybe even more. And it's interesting that for instance, like traditional Chinese medicine, they would go straight to a part of the body
Mm-hmm.
to diagnose a mental problem. And as you're
Mm-hmm.
saying it, I'm like, oh, actually, that's the first time I've thought there might be something there. Actually,
Mm-hmm. Mm-hmm.
you could,
Mm-hmm.
they might have already looked with some form of intuition or awareness at those parts of the body that are related.
Yeah, I mean, I would be very open to diagnosing what's happening in the brain or what's happening to diag to being aware of what's happening in the brain through studying the body and vice versa. Being aware of what's happening in the body by studying the brain. We can probably tell based on the changes in our emotion and energy levels. There's probably a way to tell what goes on in the body as well. It's a reciprocal connection. They both interplay with each other.
Yeah, it's like biology recently. I don't know what's happening there. I thought it was a field that was relatively well understood, but there's just some
Oh,
incredible,
there's
both
there's
neuroscience and biology.
there's so much to understand. It you know it, we always make everything neat in all of the papers and all of the textbooks. It all seems that you just, you do an experiment and it works out exactly as it is on the page. In reality, everything in biology is just so messy. All you can do is grasp at some straws of meaning in this constant torrent of chaos and try to cling on to some patterns, but I definitely don't get a sense that biology is well understood. I get a sense that most of it is, we'll never get a chance to even conceptualize what it means. And thankfully, we can pick out some patterns and just, try to hold on to them as best we can. That's my feeling of working in the lab.
Yeah, I think the more we find, the more we
The more
realize we
questions we
don't
have.
know.
Absolutely. Yeah, could not agree more.
So, how do you see this influence in like treatments for like neurological conditions or diseases, like rooted in mental or memory dysfunction?
Hmm. You mean how our studies might apply to neurological conditions?
Yeah,
Yeah, yeah. Well, I think the deepest bottom line of our study is that patterns matter. Patterns matter on a surprisingly fine scale. They matter for surprising range of cells. And I think the more we know about our body, the more attuned we will be to the patterns of input. And that means patterns of input into our brain, patterns of input into our body. What's the most effective prevention of dementias? All the things that we already know: social relationships, healthy lifestyle, walking in the park, fulfilling job. All of that we know makes you a healthier person. But we don't know exactly what that means. We don't know exactly what is a healthy diet. We don't know exactly what kinds of patterns of social relationships make you the happiest. And there's a possibility that we might find out. We might be able to tell exactly with what frequency should we be eating exactly what kind of food to get exactly what kind of emotion. I mean, at the moment, this is science fiction. At the moment, it seems crazy. But the fact that patterns matter on a finer time scale that we previously believed makes it possible that in the future we will be able to construct our experience based on what we know about how those patterns convert into what outcomes. I know it's very general at the moment. I don't have a specific technique to save yourself from Alzheimer's, but I think those techniques might exist, and it's up to us to find them out. And that's true for the brain. It's equally true for the body, in health and disease. In cancer, for example, our standard paradigm now is to kill cancer cells with chemotherapy. Chemotherapy is poison. Poison for cancer cells, also poison for other cells. The goal is to keep that level of poison at the level that would kill the cancer cells as much as the patient can tolerate for as long as possible until all the cancer is dead, and then you take that poison away. But it might be that there's a better pattern. Maybe we can achieve better killing of cancer cells by administering some non-obvious pattern of stimulation, by putting in that poison not for a long period of time, but administering it in bursts that are unevenly spaced to take advantage of the memory of those cancer cells. There's a sizable body of research that now looks at cancer drug resistance as a form of memory in cancer cells, and asks how can we make those cells forget that they are resistant to that drug so that we can kill them more efficiently. So that's an example from a devastating disease, but the same could apply in less terrifying situations: obesity, just exercise. We don't have a theory of how to time processes in our life. We do that arbitrarily. We do that based on overall impressions that more often is better, less often is good, or vice versa. But we never have a precise theory of how to arrange those events in time to maximize health and to maximize well-being. And I think our research points to that being possible. That's as much as I can tell.
Wow, that's also incredibly exciting. Is has anything been discovered about cells forgetting and how that might be done, or is that
Mhm.
also in early research?
Well, so there's a couple of different ways to think about forgetting. Could tradition and right now I'm talking about mental forgetting. Forgetting as a psychological phenomenon. It used to be thought of as almost a passive process that things happen when we memorize, but then they just passively, gradually wane and get forgotten. And that's true to some extent. There does seem to be more information processing happening at the front end of memory formation, and the back end, the removal of the memory, is sort of just this process that mills over every memory, regardless of what it is, to some extent. But we now know that there is, for example, active forgetting, that some memories are preferentially, erased when the brain decides that it needs to get rid of them. Some memories are more stable and don't just passively disappear. So you can control the process of memory formation at the level of forgetting. We understand now that forgetting is equally as important for memory as memorizing itself. You don't want to retain every piece of information that passes through your brain. It would be counterproductive. You want to generalize, you want to remove specifics and focus on the big picture. So forgetting is just as much a part of memory formation as the learning, as the front end. So is the same true for our cells? To some extent, yes. We know that there are both passive and active processes in the removal of this the cellular memory that we're studying. It will eventually just disappear, but there might be a way to accelerate the process. That is something that we're very interested in. Because if we can figure out a way to like accelerate the disappearance of that cellular memory, well, that might point to a way to, for example, clear your brain of addiction. Let's say addiction is a pathological memory that you don't want. You want to get rid of it. If there was a pharmacological way that you could tell a cell accelerate your forgetting and administer it just to your cells that is responsible for that addiction, that would be a great medical intervention. So that's something that we're interested in. It's a work in progress, early days. I can't tell you much about it, but definitely something that we're interested in. And who knows? Again, we don't yet know exactly what role body memory plays in our body. We can only speculate about that. But there probably are some body memories that we don't want. There are probably some that we want to occasionally get rid of, and accelerating that forgetting process would definitely be beneficial. So stay tuned.
Yeah, that would be, I mean, the addiction, that would be massive. And also, I assume could be applied to so many different issues, particularly societal issues. It's really
Of course,
interesting
the
you
difficulty
the
is
map.
to forget that, but not forget everything else that you remember in your brain. It would probably be easy to just wipe all the contents of the entire brain, but well, we don't want that either.
The full data, full reset, factory reset. That would
Yeah,
be.
factory reset, exactly. Yeah, yeah, yeah. That's kind of actually reminiscent to what we're currently doing with ourselves. We're trying to achieve a factory reset, basically. But it's easier with individual cells than with an entire brain.
Yeah, it's a little bit more nuanced at that
Yeah,
scale.
yeah. Hmm.
So I've heard you suggest that the brain is based on memory rather than the other way around. Could you elaborate on that perspective?
Well, in the past, people thought of memory as something that you plug into. It's something that's external to a brain. It's almost an object that you place somewhere in the brain, and there it sits. And when you access it, you reach into this object and you grab it, and you can take it from there and place it somewhere else. But it's not what memory is. It's not even just how it works. It's not what it is. A memory is not an external object to the brain. It's not a standalone object. It's not dissociable from whatever is memorizing it. If we're talking about the brain, well, a memory is how that brain changes in response to experiences. All the combination of all the changes, instant changes that immediately disappear, long term changes that take forever to clear off. All of those changes taken together, that is what memory is. You can't separate one memory from another, and you can't separate memory from what memorizes it, because you can't separate a change from what is changing. That's maybe the easiest way to put it. So that's what I mean by the brain is memories. Every single action, every single activity that happens in the brain modifies it in some way, big or small, short or long, and all of that is memory.
So, given your findings on the mass-spaced learning effect, do you see would that also would that apply to full-size brains as well, to all
Well,
learning?
that's the part that we do know. That's our starting point for this research. The mass-spaced learning effect is one of the most fundamental properties of memory that we know about. It seems to work regardless of what kind of memory this is, good or bad, or procedural memory or episodic memory, whether it forms in humans or in mice or sea slugs, or even if you study entire student classrooms and measure their performance. No matter what you look at, if you have the same amount of training, if you administer it in one crammed go, you form an inferior memory to if you administer that same amount of training in spaced intervals. That's as rock solid as anything could possibly be. It was established by Hermann Ebbinghaus, this German researcher from the 1800s who studied his own memory. He memorized these nonsense words and repeated them to himself in very specific patterns and measured how well he remembered them. His whole life was an experiment. And he established this spacing effect first. So that has been seen in brain systems, but never outside the brain. So really, the news of our work was that actually that famous psychological effect that we all know and love, actually cells have it too. Cells also follow the same rule if you give them experiences in the form of chemicals, and if you observe their memory in the form of this glowing artificial protein that they produce. And if you do that, they also form a superior memory, more of that glowing protein for a longer time, if you administer the chemical pulses in spaced intervals, rather than cramming them all in one go. So that was the big story of our study, that this rule is not a brain thing; it's a cell thing.
How does that, is there a limit for the time interval, or is there like an optimal amount? I assume you had to.
Yeah, no, of course, it's a very astute question, because it can be infinite. If it's infinite, if the time extends forever, well, then you just have individual single events. It's not a repetition. A cell has to have some peak, some moment in time when it decides that repetition is most meaningful. If it's zero, well, then you're cramming everything in one go. If it's infinitely long, then you're just doing single events on different days. But there is some optimal, we call it ITI, intra-trial interval, that produces the superior memory. And what's interesting is that we think that different kinds of signals, so different kinds of signals can induce the cellular memory, uh, produce this glowing protein that we're looking at. We can input different kinds of chemicals. All of them emulate signals from other cells, but there are different types of signals, and so we can emulate them all independently. And all of them produce some response on our glowing protein, produce this memory. But it appears that different signals are tuned to a different time interval. So some of them produce the most superior memory when the time interval is 10 minutes. Some of them produce it best when the time interval is 20 minutes. We suspect that another one produces it at much shorter time intervals. So we think that real life cellular experience is a combination of all those timelines, because the cell is not just receiving a single signal at a time, it's constantly listening into thousands of different signals. And for each of those signals, there's some optimal timing when the cell responds to it maximally. And so its overall response is some averaged out response to all of these different timelines. That's how it analyzes the patterns. By combining all those patterns of all those chemicals into a single response, it abstracts all of this timing information that is tuned to into a particular physiological state. So we think that having those different molecules that are tuned to different time intervals. It is a way for a cell to analyze complex patterns of information.
In a popular sort of contemplative research approach in neuroscience, sort of where the Eastern traditions are meeting neuroscience, I guess you could say, is that the self is a construct potentially of the brain. We spoke to Shamil Shandaria about the Bayesian brain hypotheses, and yeah, as you're probably familiar with the free energy principle
Yeah.
and how the brain appears to construct reality, and we are now applying those models to AI and
Mm-hmm.
machine learning
Mm-hmm.
algorithms. So for anyone that doesn't know, basically there's computational stacking within the brain that your brain is trying to predict reality just because it's computationally very explosive. There's a lot of data coming in from the senses and now potentially data coming in from the cells. And your brain needs to take the past information and predict the future
Mm-hmm.
because there's just not enough bandwidth in the
Mm-hmm.
brain. So, yeah, is it possible that what we call the self is also a construct of or an accumulation of kind of like accumulated memory, even cell memory?
I think it's it's very reasonable to say that. I think that what you described as prediction, this top down flow of information, that our brain uh, conceptualizing this information that's coming from the senses. Where does this top down flow of information come from? Where do those predictions come from? They come from memory. So I use this in my book and class. I use visual illusions. Well, for example, there's one with a where you can see either a young woman turned away or an old woman that's kind of in profile. It's a clever picture, but you can only see one at a time. You can't see them both at the same time. Even though you understand exactly how it works, you see where all the lines go. No matter how hard you try, you can't see both pictures at the same time. Another one is like two faces and a vase in the middle. Very simple, nothing's mysterious about this, but your perception can only focus on one or the other at that time. So that's that's the prediction. That's the We like to believe that we just passively take in senses, analyze them, and come to a conclusion. But we're actually actively participating in our perception. We're actively picking out things that we want to be perceiving. To perceive something, you have to decide that I'm either going to be looking at the faces or I'm going to be looking at the vase. You have to make that decision, and then that's when you perceive it. But that decision originates from memory. You had to see faces at some point in your life. You had to see vases at some point to memorize what they look like, to carry that image in your mind and to pick it out from this chaos of sensory experience. So, in that sense, yes, absolutely. Our self is a layering of such memories. It is a layering of predictions that it's overarching predictions that predict smaller predictions that predict even more specific predictions that predict even more specific predictions that predict what we will receive through our senses in real life. It's a nested hierarchy of levels of prediction, all of which are embedded in the memory of those cells that carry those signals around. So when we talk about cellular memories, that includes the memory of neurons. Yes, all those neurons are cells. Neurons are not different from any of all those other cells. So definitely their cellular memories contribute to those nested levels of prediction that form who we are. Are other cells of the body also involved in those nested levels of prediction? Maybe. Maybe. We don't know about that yet. That's not part of the free energy principle by Friston and Clark. But I can easily see that they are involved in those levels and they participate in either supplying the information into the brain, as we discussed, is possible for trauma, or even maybe are participating in predicting that information, in giving in producing that top-down flow of information that predicts ongoing behavior. One good candidate could be glial cells in that are inside the brain but are not neurons. They are well positioned to be non-neural cells that could retain some imprint of experience and could control the behavior of neurons responding to ongoing information. And so, yeah, easily the state of a glial cell could contribute to our sense of self. I wouldn't be surprised by that.
Wow, we would need there would be big updates with the model of our understanding of the
Mm,
world if that was the case. And yeah, as we said earlier, I think the more we the more we discover, the more we realize we don't
mm,
know.
mm. Of course, the more we realize we still have to learn. I mean, we're now at the point when we are beginning to, well, I would say what AI now does is maybe not emulates, but it is at least inspired by the electrical behavior of neurons. But this electrical behavior, those electrical signals that are passing between neurons, that is only one aspect of what they do. That is only one aspect of their physiology. Uh, there are thousands of other aspects of thousands of other processes that are happening in neurons that are not electrical pulses. And so I think that as we progress in technology and artificial intelligence, we will start incorporating more and more of those other neuronal biological processes and cellular processes into our models of intelligence. Right now, it's all electrical pulses interacting with each other. But if we incorporate more cell-like behaviors, then our AI might become more biorealistic. It might have a more realistic memory, for example. It might respond to patterns of input more like a human would. It would learn from them in similar ways. It would modify itself, would be more flexible, maybe. So I would say that if I want to predict the direction in which AI will go, I think it will gradually become more and more similar to the body and to cells specifically.
Yeah, and then you've also got like Penrose and Hammerhoff over there trying
Oh yeah,
to
with the microtubules.
yeah, point
Yeah.
at potential fundamental consciousness. It's just yeah,
Mm.
it's oh, science has just become so interesting recently. I've also been following, yeah, Professor Michael Levin's work pretty extensively, and it seems like there would be a convergence, and that it's about to or is already connecting pretty well with your work.
Mm-hmm.
He's looking at bioelectricity with pattern memories for development and regeneration.
Mm-hmm.
So is that something that you have been looking into as well? And what's your
I'm very
understanding?
inspired by Mike Levin's work. He's definitely a major role model for me. I look up to him, and he's given me good advice, and we've shared our studies. We've sent them our cell lines. Hopefully, they will do something interesting with them as well. Um, yeah, I think our biggest point of overlap with Mike Levin is that we both think of these cellular processes like memory, like cognition, um, not metaphorically, but literally. And that's a big part of our message to the world, that when we're saying cellular memory or cellular cognition, as we already discussed, we're not using these words in quotation marks. We mean them literally. We mean that the more advanced forms of cognition and memory are built upon these molecular tools that have been around since before there were brains and animals and humans and anything like that. So all of these processes have deeper evolutionary roots than we like to believe. On that point, we truly agree, and I think that our work is supportive of each other's in that respect. As far as bioelectricity, and well, I mean, anything that passes through a neuron spikes as bioelectricity. So in a sense, I am studying bioelectricity. We haven't touched regeneration yet, so I would say that Mike Levin's work uh, is more on the body side. We, because we're coming from the brains, we're more still on the neuroscience side, but we're working on very, very similar problems and definitely like-minded researchers.
Amazing. Could you briefly describe what his fundamental findings have been that have surprised you?
What his fundamental findings are? Well, I mean, there's a lot of research that comes out of Mike Levin's lab. A lot of it is theoretical. I would say his most fundamental findings are that cells are not driven exclusively and maybe not primarily by their genetic constructions, but instead their life is rich in communications with other cells in the body, and that communication is what sculpts our organism as it is. So we used to believe that everything in our body is given to us in those 20,000 genes in the sperm and an egg, and everything else is just this program unfolding in time, all those instructions unfolding in time. But actually, through communication channels such as bioelectricity that used to be ignored but now turned out to be information rich, cells can organize complex modes of communication that supplement and really build upon those genetic instructions given to us in evolution. So the way that it used to be thought of, we have genes, that sets up the body, and then on top of that we have the brain, and that learns from experience. But what Mike Levin's work says is that all cells learn from experience, and that's most of what the organismal biology is. The very setup of our body, our very multicellularity, is cells learning from experience and organizing this body through communication with each other. So I think that's the deepest philosophical message of Mike Levin's work. And what was so surprising for me? Well, I thought that electricity was exclusive to neurons, and that whatever happens in outside cells, it maybe is, it maybe affects their state, but it's not something that is information rich. Basically, I think in both his work and in our work, I'm surprised by how smart non-brain cells really are. We used to think of them as dumb, but actually there can be, I should say, almost as smart as neurons. Okay, neurons maybe are a little bit smarter. Neurons operate on millisecond timescales that other cells maybe don't quite reach, although who knows? We'll see.
Just to circle back to an earlier question, yeah, is that something that you think about much in terms of consciousness being fundamental? If cells are storing memory, I mean, the next question would be where is that where
Mm-hmm.
is that stored? And then at the same time, we have all the consciousness research happening and seemingly some evidence coming from Penrose and Hameroff that at least the microtubules are like heavily connected to consciousness. In the, I think if we have anesthesia, my understanding is they
Mm-hmm.
modified an fMRI scanner, and I don't understand this well enough to be quoted on it, but that they could see that the microtubules were functioning less, or um, and on the other side of that, subjectively consciousness was being shut off. So yeah, is that something that you've put much thought behind, or is that a little bit further down the path as well?
No, I think about consciousness a lot, and I'm aware of the microtubular hypotheses, theories. Can't say I completely understand them. I'm also a little bit scared of all the microtubule people because they're very aggressive for some reason. I don't know, people who believe that consciousness is in microtubules. God forbid you say something against that. Oh my God. So, what I don't understand about those microtubular theories is, from what I understand, most of them are based on the fact that microtubules have quantum properties. They have many degrees of freedom, and they can store their state. They can form memory. You can store memory in a microtubule, and that they are important for the functioning of the brain. I think all those three things are completely true. I don't disagree with any of them. But I don't know if that necessarily leads me to believe that microtubules carry consciousness. I think it's a big leap from saying that consciousness involves some quantum scale processes to the content of our consciousness is determined by quantum mechanics. I think that's a big leap. Everything involves quantum processes. Any chemical reaction involves quantum processes. So simply saying that there are some quanta flying around, that the quanta are involved, is not really saying anything. I think what is attractive about quantum mechanics with regards to consciousness, and I think that's where Penrose originally comes from, is this idea of a collapse, of the wave function collapse, because quantum mechanics tells us that particles exist in this ambiguous state. They're neither here nor there. They're neither a particle nor a wave. They are spread. Their probability of existence is spread around this, electron's probability is spread around the nucleus of an atom. And when you try to detect it, when you try to place a finger in it, when you try to put it into a machine that detects it, that's when it collapses into here or there. And so it seems that a similar thing is happening in our mind. It seems that interpretations of reality exist in this ambiguous state, and when you try to understand something, when you try to become aware of something, you are searching for ways to collapse that ambiguity into specific states, and it might resolve one way or it might resolve another way, and that's when you become conscious of something. I think that is a good metaphor. I think it's an analogy that makes sense in the same way as a wave function collapses into different states of the particle, in the same way the network of our brain, the network activity of our brain can collapse into specific states of that brain. And in that sense, sure, it's similar. But I don't think it necessarily means that wave function collapse is at the heart of the collapse of this neuronal network into specific states. So I think that's why quantum mechanics is so attractive as an explanation for consciousness. So I simply don't see a need to invoke quantum mechanics to explain conscious processes. This need might arise in the future. And I'm willing to accept that there's something about wave function collapse that is critical to the content of consciousness that I don't yet understand. But I don't yet know what that something would be. So I don't see a necessity for invoking quantum explanations in consciousness. What puzzles me about microtubules in memory, if memory really is stored in microtubules, that I don't understand how we explain, for example, the effect of protein synthesis inhibitors on reconsolidation of memory. So long-term memories are known to become malleable when you think of them. When you recall a memory, it turns into this temporarily malleable state, and during that state you can disrupt that memory if you give a protein synthesis inhibitor. So if you give a person that protein synthesis inhibitor, well, you shouldn't be giving it to a person because they're toxic. Let's say a rat. If you give a rat that protein synthesis inhibitor without it thinking about the memory, just at a random time, nothing will happen to that memory. But if you give it while it's thinking about that memory, as it's actively recalling it, and then you give that protein synthesis inhibitor, and it will forget that memory. So in that moment, it becomes flexible, and you can disrupt it by blocking the synthesis of proteins. If the memory is stored in a microtubule, why does that happen? A microtubule is already there. They don't need to get resynthesized at that time frame of a couple of hours when you would administer that drug. So that to me is the biggest question mark in memories being stored in microtubules. But memories are not stored in any particular place. Memory is a stable state of a cell, of a molecule, of a neural pathway. For whatever reason, the cell has has left its configuration where it was before, and now it's stabilized itself into a new configuration. Where is this configuration stored? It's not stored in any particular molecule or any particular gene. It's stored in the configuration as a whole. Everything is different and nothing specifically. That's true for the cell. That's true for the brain. That's true for any vehicle of memory. And is a microtubule part of that configuration? 100%. I don't doubt that. I'm sure that microtubules store some of the changes that happen to brain cells or to other cells during experience, and that those changes are meaningful to what that cell does in the future. I just don't see a reason to privilege microtubules in such a way as some people do it. There's lots of other molecules in the cell that I love equally as much, and I think are equally wonderful and could store many different aspects of experience in equally sophisticated ways.
And it looks like there's already enough to discover in the near future right in front of you. We probably don't, yeah, potentially need to make that leap. But I agree. Yeah, I see how they might start to make that leap just from, I mean, consciousness has been struggled with for a long time. And
Mm-hmm.
now we have quantum mechanics, in some ways, has also been struggled with for a long time.
Yeah,
But
it's attractive to
equally as
connect
weird.
those two mysterious things, and I think it also somehow subliminally it elevates you. You know, we want to be mysterious. We don't want to be just dull electrical signals. We want to be, another dimension of reality, and I think it's just an attractive thing. But I don't know. I'm okay with being just electrical signals.
Yeah, I mean, it's already mind blowing,
Yeah,
particularly
it's
with your
complicated
your
enough. It's
recent
not
discoveries.
insulting to me.
So, yeah, we mentioned earlier your incredible book. Um, I'll repeat it again, but it was one of the most beautiful book covers I've ever seen. That's upcoming in October, I think you said. So, yeah,
Yeah.
could you tell us a little bit about that, the title that you've landed on, and
Yeah.
what it's about?
So the book is called One Hand Clapping: Unraveling the Mystery of the Human Mind. And one hand clapping, as you I'm sure know, refers to a Zen koan. A koan in Zen Buddhism is a riddle. It's an unanswerable question. It's a riddle without a solution. And so in this book, in the same way as this koan asks a seemingly unanswerable question of how could a single hand produce the sound of clapping, in the same way I ask the question of how could a human mind exist in this world of atoms and molecules? I had a student come to my office a few weeks ago, and I think she asked me a question, and I was like, "This is what my book is about. I've been trying to phrase it for years now, and you've just captured it in one question." She asked, "You know, it, if life is all atoms and molecules, and if we are just animals, then why does it feel so meaningful to be myself?" That's really what the book is about. If we're in a science class, it seems that the world is made of the same stuff. Everything is made out of the same atoms and molecules, and chemically speaking, I'm pretty much equivalent to a mushroom, and even my neural organization is not that different from a mouse or a fruit fly, and yet it feels fundamentally different to be me. It feels different to be me versus all the other humans. I have my own consciousness, and it feels very different from looking at everybody else in third person. In the same way, it feels different to be a human compared to all those other animals. Even though science tells us that there is no better or worse evolved, that we are all evolved in the same way, we're equally developed, and for some reason it feels different to be a human. It feels different to be alive versus being not alive, even though we're made out of the same atoms. So where does this specialness come from? Where does this meaningfulness come from. And so it really is the same story. It is the same case of specialness that we can trace from the very origin of life, through the birth of the cell, through the appearance of animals, and then vertebrates, and then mammals, and then primates, and then humans, and then our own self. It is the same common thread of I am special compared to everybody else. And so in the book, I try to understand, what does it mean to be me from the perspective of the world around me? What does it mean to be a human, a conscious individual here now, from the perspective of the entire universe, life on Earth as a whole? That's what the book is about.
It sounds incredible, and I look forward to getting a copy. It's already out in Russia, I believe. How was
Yeah.
it received there? I believe
Yeah,
it
it
was.
did really well in Russia. So it was published a few years ago, and it won multiple awards and still selling very well. I would say that the Russian version is much thicker than the English version. It's a lot denser. I think that Russian people have more tolerance for longer form content, and it took me a while to not even translate or even rewrite, but to rethink the entire book from the perspective of an English speaking audience. It wasn't really anything specific. It was a wonderfully interesting experience of just re-experiencing it in a different language. But yeah, it's still recognized and still sold in Russia, and I'm proud of that.
Yeah, amazing, amazing, great work. And if anyone wants to find out more about you or your work, where's best to find you?
Yeah, you can follow me on social media, Nico_Kukushkin on X. You can go to my website, NikolayKukushkin.com, for all the links to all my writing, my book, my press appearances, and everything else that you might want to know about me. Everything's on the website. So I would say either one of the two, Twitter or my website.
Amazing, and we'll pop the notes below. Oh, thank you so much. I really, really enjoyed the conversation, and yeah, keep going. I think you're doing incredible work, and
Thank you,
I'm really
Josh. It was a lot
excited
of fun. Thanks so much.
to see what happens in the future for you.
Yeah, I'll keep you posted.
Thank you so much for listening to the episode. If you enjoyed it, please, if you could give us a like, a subscribe, and hit the notification bell, that would really help us out. Or just share this episode with a friend, share the course with a friend. Anything would go such a long way. Thank you so much, and I hope to see you soon.